If you had to pick one idea that makes multicellular life possible, stem cells would be near the top. A stem cell is not just a cell that can divide. Many cells divide. A stem cell is special because it can do two jobs that usually pull in opposite directions. First, it can self-renew, meaning it can make more stem cells like itself over long periods. Second, it can generate specialized descendants, such as blood cells, skin cells, or other functional tissue cells. That two-part identity is crucial. If a cell only specialized, your stem cell pool would disappear quickly. If it only self-renewed, you would not get the diverse cell types needed to build and maintain an organism. Stem cells are therefore both a biological origin point and a maintenance system. They are often described as undifferentiated or only partially differentiated because they have not fully committed to one mature role. In a lineage, they sit upstream, while progenitor and mature cells sit downstream. And that distinction matters, because it explains why stem cells are rare, heavily regulated, and so powerful in both development and medicine. To understand stem cells, it helps to understand differentiation as a one-way path with branching routes. Early cells in development begin with broad potential. As signals guide them, they commit step by step, narrowing what they can become. Stem cells are at the earlier stages of that branching map. But even within the stem cell category, not all cells are equal. Some can become nearly any cell type in the body, while others are restricted to a smaller family. Scientists use terms like pluripotent, multipotent, and unipotent to describe that range. Pluripotent means broad potential across many tissues. Multipotent means several related cell types, often within a specific system. Unipotent means one specialized output, though the cell may still self-renew. This spectrum is important because public discussions often treat stem cells as one thing, when in reality stem cell function depends on developmental stage, tissue context, and molecular environment. The practical lesson is simple. When someone says stem cells can become anything, ask which stem cells, under what conditions, and in what biological setting. Embryonic stem cells represent one of the most potent stem cell states known in mammals. During early embryonic development, around the blastocyst stage, a small inner cell mass contains cells that have not yet committed to final tissue identities. In living development, these cells will eventually contribute to all major body cell types. Their natural path begins with early patterning events that form the three germ layers, ectoderm, mesoderm, and endoderm. From those layers come the organs and tissues of the body. What makes embryonic stem cells scientifically remarkable is that when they are isolated and grown in carefully controlled lab conditions, researchers can maintain them in a stem-like state rather than allowing full differentiation to proceed. That creates embryonic stem cell lines, which are stable research tools. These lines let scientists study developmental pathways, test how genes influence fate decisions, and attempt directed differentiation into specific cell types. So embryonic stem cells are not just powerful because of what they can become, but because they let researchers observe and guide the earliest decision-making logic of human biology. Adult stem cells are different in mission and design. Instead of building an entire body from scratch, they preserve and repair existing tissues across life. They live in protected microenvironments called niches, often in specific anatomical locations like bone marrow or reproductive tissues. A niche is not just a place, it is a regulatory system. Surrounding cells, extracellular matrix, local chemistry, and signaling molecules all shape whether a stem cell stays quiet, self-renews, or differentiates. Adult stem cells are usually more limited in potential than embryonic stem cells, commonly multipotent or unipotent. That makes sense. Biologically, your bone marrow does not need to suddenly generate neurons, it needs to produce blood and immune lineages reliably. Adult stem cells are also relatively rare compared with progenitor cells and mature cells. In practice, much of day-to-day tissue turnover is handled by downstream progenitors, while stem cells act as a strategic reserve that replenishes the system when needed. This hierarchy balances stability, efficiency, and long-term resilience. A concrete example is hematopoietic stem cells, often abbreviated as HSCs. These cells reside mainly in bone marrow and sustain blood formation across a lifetime. Every day, your body needs massive numbers of new blood cells, oxygen-carrying red cells, multiple immune cell classes, and support cells involved in clotting and defense. HSCs sit at the top of that production tree. They self-renew, then generate progenitors that progressively specialize into different blood lineages. This system demonstrates why stem cell biology is not abstract theory. It is operational infrastructure inside the body. Similar principles apply in other tissues. Basal stem-like cells support skin renewal. Mesenchymal stem cell populations contribute to maintenance of connective tissue components such as bone, cartilage, muscle, and fat. In each case, stem cells are not working alone, they are integrated into layered production pipelines. Understanding those pipelines is essential for medicine, because therapies can fail if we ignore where in the hierarchy intervention should occur, at stem cells, progenitors, or mature cells. Modern stem cell science did not appear overnight. A major foundation came from work in the 1960s by Ernest McCulloch, James Till, and Andrew Becker in Canada. Their experiments provided strong evidence for cells in bone marrow that could both self-renew and generate differentiated blood colonies, helping to establish the functional concept of stem cells in a rigorous way. This was a turning point because it shifted discussion from speculation to experimentally testable cell behavior. Over time, advances in cell culture, transplantation methods, and molecular tools expanded the field. But history also reminds us that stem cell progress has been uneven. Basic discoveries often came years before clinical applications. That lag is normal in biomedicine. A mechanism discovered in a lab model must survive replication, scaling, safety testing, and regulatory scrutiny before becoming routine care. So when evaluating headlines, historical perspective helps. The field has real achievements, but it also has long translation timelines and frequent gaps between laboratory promise and standard medical practice. One area where stem cell therapy is clearly established is hematopoietic stem cell transplantation, first performed in the late 1950s and developed over decades into a core treatment framework for selected blood disorders and cancers. In this approach, the goal is to rebuild a patient's blood-forming system after disease or high-intensity treatment. Conceptually, it is an elegant use of stem cell biology, replace or rescue a damaged production source rather than only treating symptoms downstream. Its success also sets an important benchmark. It shows stem cell medicine can work profoundly when biology, clinical indication, and delivery method align. But it does not automatically validate every other proposed stem cell intervention. Different tissues face different challenges. Cell survival, integration, immune response, and long-term behavior can vary dramatically. So this established therapy is both an achievement and a caution against overgeneralization. It proves the principle while highlighting that each new stem cell application must be judged on its own biological and clinical evidence. In the late 1990s, researchers succeeded in deriving and maintaining human embryonic stem cell lines, opening a new era for developmental biology and regenerative research. This offered unprecedented access to pluripotent human cells in vitro, making it possible to model early differentiation events and attempt directed generation of specific tissues for study. However, the same step triggered intense ethical debate because obtaining these lines traditionally involved embryos at early developmental stages. Different societies responded differently. Some jurisdictions imposed strong restrictions, while others supported regulated research. These policy differences shaped global research geography, funding patterns, and training pipelines. The controversy was not only moral, it was also practical, affecting who could do what science and where. For viewers, the key point is that stem cell science sits at the intersection of biology, ethics, law, and culture. Scientific capability alone does not determine research trajectory. Social values and regulatory frameworks co-author the timeline of what becomes possible. A major shift came in 2006 with induced pluripotent stem cells, or iPSCs, pioneered by Shinya Yamanaka's team. The central idea was radical and simple at once. A mature body cell is not always locked permanently into its identity. Under the right molecular instructions, it can be reprogrammed back toward a pluripotent-like state. That means researchers can start from differentiated cells, such as skin-derived cells, and push them into a stem-like condition without using embryonic material in the same way as traditional embryonic stem cell derivation. This does not erase all ethical questions, but it changed the landscape dramatically and expanded research options. iPSCs also enabled patient-specific modeling, where cells from an individual can be used to study disease mechanisms in a controlled lab system. It reframed cell identity as more plastic than once assumed and helped launch an era where reprogramming, not just isolation, became a central strategy in regenerative science. Even with iPSCs and advanced culture methods, translating stem cell concepts into robust therapies remains hard. Scientists must control differentiation precisely, because mixed or unstable cell populations can undermine safety and effectiveness. They must ensure cells behave as intended after transplantation, including long-term stability rather than short-term improvement only. They must manage immune compatibility, integration with existing tissue architecture, and functional performance in real physiological environments. And they must do all of this under strict manufacturing and quality standards. This is why claims of quick stem cell cures should be treated cautiously unless backed by solid clinical evidence. In parallel, stem cells are already transforming medicine in less visible but highly meaningful ways, such as disease modeling and drug screening. These uses can improve understanding and treatment development even before direct cell replacement becomes routine for many conditions. Progress in this field is real, but it is usually incremental, evidence-driven, and dependent on disciplined biological control. Another important distinction is between stem cells and neighboring categories like progenitor or precursor cells. In public discourse, these terms are often blurred, but biologically they describe different capacities. Progenitor cells can proliferate and generate specific lineages, yet they typically have more limited self-renewal than true stem cells. Precursor or blast cells are often even more committed to a particular fate. Why does this matter? Because therapeutic strategy depends on it. If you need durable, long-term tissue maintenance, stem cell-level self-renewal may be necessary. If you need rapid short-term replenishment in a defined lineage, progenitor cells may be more appropriate. Confusing these categories can lead to unrealistic expectations and poorly designed interventions. A useful mental model is to think of the cell hierarchy like an organization. Stem cells are long-term leadership and continuity, progenitors are operational managers, and mature cells are specialized staff executing specific tasks. Effective medicine needs the right level in the hierarchy for the problem being solved. So where does this leave us? Stem cells are not magic, but they are foundational. They explain how complex organisms develop, how tissues renew, and why regeneration is possible at all in many systems. Embryonic stem cells reveal broad developmental potential. Adult stem cells sustain day-to-day biological continuity within specific tissues. iPSCs demonstrate that cellular identity can be rewritten under defined conditions, opening pathways for research and, potentially, future therapies. At the same time, ethical frameworks, regulatory systems, and technical limits shape what advances reach patients and when. The most accurate view is neither hype nor cynicism. Stem cell science is a mature, high-impact field with proven successes in selected contexts, major unresolved challenges in others, and extraordinary value as a lens into human biology. If you remember one thing, remember this: stem cells are the body's long game, balancing stability and change across a lifetime, and teaching us that identity in biology is structured, dynamic, and, in some cases, reversible. If you found this helpful, please like this video and subscribe for more explainers.